Hello everyone, I hope you are well. So this video is going to be about antibiotics, and I'm planning on doing two parts. So part one is more about the different classes and the way the antibiotic works, and part two is going to be the spectrum of activity and side effects. So here's a cell and a bacterial cell, and this is just a very simplified cell. Okay, so the first thing, we have our cell membrane, which is gonna be this part. Okay, think of this part. Okay, then the peptidoglycan layer, which is gonna be this outer yellowish color, and then we have our nucleus, which is over here, and the protein synthesis that takes place over here. Okay, so yeah, that's just a very simplified cell, bacterial cell. Okay, so we're going to use this cell and explain on how the antibiotics work. So first, I mentioned the peptidoglycan. So first I want to talk about the peptidoglycan itself. Okay, so the peptidoglycan, so what it is it's complex polysaccharides. Okay, so it's complex polysaccharides, and it consists of two types of sugars, a NAG and a NAM sugar. Okay, so and they are their structure is very similar to glucose. So the peptidoglycan layer, it is complex polysaccharides that consists of NAG and NAM sugars. Okay, so they are linked together to form a chain. Okay, so it's NAG and then it alternates with NAM, and then that chain, that's gonna be the glycan part. Okay, of the peptidoglycan. So they cross link or cross bridge, okay, 4 amino acids, so they're tetrapeptides. Okay, so they cross link with each other, and then they form the peptidoglycan layer. And then depending on the bacterium, the type of bacteria, the tetrapeptide bridges, they are covalently bonded to one another. Okay, and then they are held together by a short connecting chain of other amino acids. Okay, then you get gram positive and gram negative. Now the only difference is the peptidoglycan layer. Okay, so for gram positive is going to be a thick peptidoglycan layer, and for gram negative they're going to have a thin peptidoglycan layer. Alright, and if you want more information about this, then you can go to the resources that I link, that's going to be at the end of the video. You can go to the resource and find that on page 65. Okay, then let's start with the first type of classes, or the way they work. Okay, so we have cell wall synthesis, that they're going to inhibit that. Okay, so they are going to inhibit the cell wall synthesis. So first thing, it can be done in two ways. Okay, first is they're going to decrease the peptidoglycan synthesis, and the drugs that's going to do that is going to be your vancomycin and phosphomycin. Okay, and then they can decrease the cross linkage of the peptidoglycan. Okay, so in the previous slide we talked about the synthesis, which is the NAM and the NAG molecules, and then the cross linkage. Okay, but we'll come to that in more detail when we look at the bacterial picture again. Okay, so let's then go to the next slide. So penicillins. So here we have 3 different types of penicillins. Okay, so we have your natural penicillins, and that's going to be your penicillin G or your benzyl penicillin. Okay, and then you can have penicillin V, which is the phenoxybenzyl penicillin. Okay, So that's going to be your 2 natural penicillins. Then you have anti-staphylococcal penicillins, okay? That is going to be your oxycycline and decloxacillin, okay? And then we have aminopenicillins. That's going to be your amoxicillin and your ampicillin, okay? And then we have beta-lactamase inhibitors, but first let me explain why they add that to the drugs, to the penicillins. So when the bacteria get resistant to these penicillins, okay, so they're also called beta-lactams, okay, remember that. So they're also called beta-lactams. But when they get resistant to this, they they form an enzyme called beta-lactamase, okay. They form this enzyme. Now what this enzyme will do, it will bind onto the beta-lactam ring of the penicillins and the beta-lactams, and it will render the drug ineffective, okay. So that's what it does. So when people decided they want to inhibit this enzyme, they developed this beta-lactamase inhibitors. That's going to be your clavulanic acid. That's going to be added to the amoxicillin, okay. So it's a combination, and the tazobactam is going to be added to the piperacillin, okay. So what this will do is it will inhibit that beta-lactamase, and then it will we can say it will keep it busy, okay, while the penicillin do its job, okay. So we can sort of say that. So that's why they give the beta-lactamase inhibitors, okay. Yeah, so that is the different types of penicillins. As you can see, there's a lot. And then we move to cephalosporins. So here we have 5 generations, okay. So generation 1, we have cefadroxil, cefalexin and cefazolin. Now this cefazolin, they can usually give it for prophylaxis in surgery. Then generation 2, we have cefuroxamine and cefoxitin, and then we have generation 3, which is all of these ones. Now these ones we can say are the main workhorses of the cephalosporins, okay. Then we have the generation 4, which is the cefepime, and the new one, the generation 5, and then the carbapenems. So carbapenems, you can remember them by DIME, okay, DIME, as you can see there. DIME for doripenem, I for imipenem, M for meropenem, and E for ertapenem, okay. So as you can see, all of them end in penem. And now these ones, they have a broad spectrum of activity. So they are very, very strong. So you can just remember them by DIME. Okay, so DIME. Then when we move on to the cell, so we said that peptidoglycan synthesis, okay. So that's going to be your vancomycin and your phosphomycin. So it's gonna, let's take this, let's just highlight where we are talking about this part, okay. So that's gonna be your peptidoglycan layer. So if you're gonna inhibit the NAM and the NAG from making peptidoglycan, you're not gonna have a peptidoglycan layer. That is gonna decrease the and it's gonna make the cell weak, okay, so that things can go into the cell. And if you, let's talk about the other one that decrease the cross linkage of the peptidoglycan layer. So let's say, for instance, we do this, okay. Let me just draw this and another one. So let's say it cross links like this, okay. So let's say this is a NAM molecule, this is a NAG molecule, then a NAM, then a NAG, a NAM, and a NAG. Okay, so it cross links like this, and then it forms this layer. Okay, so let's if you're gonna decrease the cross linkage, so you're gonna break this, is it gonna form a peptidoglycan layer to protect the bacterium? No. So that's how the cell wall synthesis inhibitors work. Okay, so just to go back, all of the cell wall synthesis inhibitors, they are going to be vancomycin, fosfomycin, penicillins, cephalosporins, and gabapenums. Okay, now just important thing that all of these ones they are beta lactams. Okay, beta lactams, because they have a beta lactam ring in their structure. Okay, so that's just something to know. Okay, then when we move to the next type of mode of action, which is going to be your cell membrane integrity. Okay, so these two drugs, daptomycin and polymyxins. Okay, now what they're going to do is they're going to decrease this, they're going to play with the membrane integrity. Okay, so they're going to have an effect on the membrane and its integrity. So when we go to this picture, so we are talking about this part over here, the cell membrane. Okay, so they're gonna make it more porous. Okay, so if you're gonna make it more porous, more other types of electrolytes can go out of the cell and in of the cell, and that can cause electrolyte imbalances, which can also be problematic for the bacterium. Okay, then it can't function properly. So that's basically what these drugs are going to do, the daptomycin and the polymyxins. They're just going to have an effect on the integrity of the cell membrane. Then if we move on to the next class or the next mode of action, it's going to be your folic acid pathway, or your folic acid inhibitors, if we can say it like that. So we have two drugs. We have sulfamethoxazole and trimethoprim. Okay, so sulfamethoxazole, it's going to have an effect on the first step of the folic acid synthesis. Okay, and trimethoprim is going to have effect on the second step. Okay, and when they are combined together, they are called Bactrim. Okay, so that's just the trade name is Bactrim. Okay, so the way that they are going to work. Okay, so let's do it on this side. So there's gonna be something called PABA, which is para-aminobenzoic acid. Okay, that is gonna get converted into DHF, which is going to be dihydrofolate. That dihydrofolate is going to be converted into THF, which is going to be your tetrahydrofolate. That's going to be your folic acid. Okay, so, and folic acid is very important for protein synthesis and also for DNA synthesis as well. Okay, so like we mentioned for the first step, so for this part over here, sulfamethoxazole is going to inhibit this part. If you can inhibit that part, there is a decrease in the DHF and a decrease in the THF. Okay, so that's going to be bad for the cell. And then for the second one, that's the drug that's going to affect the second part, is going to be your trimethoprim. Okay, so this is, let's make number 2 over there. It's going to inhibit this part. Okay, if that's going to happen, there's going to be a decrease in THF as well. Okay, so now let's say you have a double decrease in THF. Okay, is that going to be good for the cell? Is it going to do protein synthesis and DNA synthesis? No. so it will inhibit these processes within the cell. So this drug, it's not gonna kill the drug, so it's not gonna be bactericidal, but it's gonna slow it down, so it's gonna be bacteriostatic. Okay, then the next mode of action is going to be DNA integrity. Alright, so drugs that we're going to use for this is going to be metronidazole and nitrofurantoin. Okay, so what they are going to do, if we go to the bacterial cell, so now this drug is DNA integrity. Okay, so we're just gonna focus on the nucleus for this one. So what it's gonna do is gonna increase the reactive oxygen species. Okay, now that's gonna be things like peroxides, superoxides, and hydroxyl radicals. Okay, so now when you increase both drugs, right, they're gonna increase the reactive oxygen species. So if that's gonna happen, it's gonna damage the DNA. Okay, so if it's gonna damage the DNA, then it's gonna affect the integrity of the DNA, and it's gonna be bad for the bacterial cells, so it can't replicate. Okay, for DNA synthesis. So that's how the metronidazole and the nitrofurantoin works. Okay, so it's going to increase the reactive oxygen species, damaging the DNA and affecting its integrity. Then it's going to affect the other processes of the cell as well. So if we move to the next class or mode of action, we have mRNA synthesis. Okay, so drugs is going to be rifampicin or rifabutin. Now these drugs, they are mostly used in TB. Okay, so they are mostly used in TB. So what's going to happen is that let's focus again on only the nucleus. Right, so what's gonna happen is for DNA synthesis to occur, or even protein synthesis, okay, there's transcription. So it makes mRNA. The mRNA, let me draw mRNA my best. Okay, so this mRNA, it's going to be synthesized in here. Okay, it's going to go into translation over here. So this is translation, and this is transcription over here. So what's going to happen is there's an enzyme called RNA polymerase. Okay, now this enzyme makes the mRNA. So what's going to happen is the rifabutin is going to inhibit the enzyme. Okay, then is transcription going to take place? No, it's not going to take place. That means there cannot be any protein synthesis and DNA synthesis. Okay, then if we move to the next mode of action, we have DNA gyrase function. Okay, so now with the drugs we are going to use is the class fluoroquinolones. There's first generation and then there's a second generation. So the first generation is going to be your ciprofloxacin, and your second generation is going to be your levofloxacin and your moxifloxacin. So they are the fluoroquinolones. Okay, so for the fluoroquinolones, how they are going to work, again we're just going to focus on the nucleus. Okay, so what's going to happen is the DNA gyrase. Okay, let's make it T. It's called topoisomerase. Okay, so if we make a T for topoisomerase, let's say it has one hand and another hand. This hand, let's say this hand is a scissor. Okay, so it cuts, and this hand is glue. Okay, so what's gonna happen on this nucleus, look at the dark part and then the lighter part. Okay, so let's say the dark part is DNA that is super coiled. Okay, so super coiled here, let's say it's gonna look like this. Okay, there's gonna be a lot of DNA stuck together. Okay, that's a very bad DNA, but you get the idea. Okay, so it's like super coiled. So what's gonna happen is this topoisomerase guy, it's gonna cut the DNA. Let's say it cuts it there and there, just for the DNA to relax a bit. Okay, then it's gonna take this other hand, the glue hand, it's gonna glue it back together. Okay, so let's make a correct mark. It's gonna glue it back together. So what this fluoroquinolones does for this enzyme, it's gonna stimulate this hand that's gonna cut. Okay, that's gonna cut the DNA, and it's gonna inhibit the hand that glues it back together. So now think this guy, it's just gonna go into the DNA over here, and it's gonna cut. Okay, and you can imagine that that's not good for anyone or anything. Okay, so that's DNA synthesis, and yeah, that's gonna be affected. So the bacteria, it will then die because of that. So it's gonna affect the topoisomerase. Okay, then when we go to the next mode of action, we have our 50S ribosomal subunit. So it's gonna inhibit the 50S ribosomal subunit. So drugs we have is macrolides, so it's gonna be these guys. Okay, so erythromycin, azithromycin, and clarithromycin, that's gonna be your macrolides. Then we just have another group of drugs that does the same thing. We have clindamycin, chloramphenicol, and linezolid. Okay, or linezolid. So that's the drugs that's gonna affect the 50S ribosomal subunit. So if we go to the bacterial cell, this is gonna be your 50S ribosomal subunit. So where the protein chain forms, this is the protein chain. So if you're gonna inhibit this part, then you're not gonna form any proteins. Okay, yeah, so that's basically just those drugs. It's a very short one. And another one is going to be your 30S ribosomal subunit. So the drugs that's going to affect that is going to be it's going to inhibit, remember that it's going to inhibit the 30S ribosomal subunit. So the drugs is going to be the class, the aminoglycosides. This is going to be your amikacin, gentamicin, and streptomycin. Okay, and then you have another class called the tetracyclines, which is going to be your doxycycline. Okay, then if we go to the bacterial cell, it's going to be this little part, the small part over here. Okay, if that's going to be inhibited, then it's going to be hard for the mRNA, which is this blue string, to anchor and make proteins. Okay, so all of the things, it's going to affect protein synthesis. Okay, so this is going to be a 30S ribosomal subunit inhibitors. It's going to affect protein synthesis as well as your 50S ribosomal subunits. Okay, inhibitors, that's gonna, all of them is gonna affect protein synthesis. All right, and then the last slide, which is just gonna be my resources. So I used Bauman and of course the SUMF. So that's what I used for this video. Thank you, and I hope you enjoyed.